Hi guys, so it doesn't matter how fast the sperm is, for it to be liable for fertilization, it must undergo two important processes, which is capacitation and acrosome reaction. So in this video, we're going to be talking about these two processes. All right, so before the process of capacitation and acrosome reaction occur, ovulation must have taken place. That means an egg must have been released into the ampulla or the fallopian tube, waiting for fertilization. And also, the sperm must have been released. And you also need to know that the sperm actually does not come alone. It doesn't come just as a cell. It has to sit in a medium, a transport medium. And that is what we call the semen. And the semen is made up of a lot of components, ranging from the energy source, fructose, fructose citric acid, as the energy source in the semen. The next thing you can find in the semen, or another thing you can find in the semen, is the prostaglandins. Now, this prostaglandins helps mobility. It helps the sperm to be mobile. You know, as the sperm is going through different parts of the female reproductive system, it will encounter different restrictions, different enzymes, different fluids. Okay, so these prostaglandins helps it to be mobile. All right, so the next thing we are going to look at is that is found in the semen is the coagulant. All right, so when the semen is released, it doesn't just scatter. It sits. Remember, I said it sits in the sperm sits in semen. Okay, so when the semen is released, there is what we call an anticoagulant. Rather, what we call a coagulant. That coagulant is most time made up of fibrin. Okay, now that coagulant just holds the sperm in place, but this particular coagulation does not last long. It doesn't last for a long time. So what happens is that it just lasts for around about 10 to 15 minutes and it goes away and the sperm starts moving. All right, so the next thing, or another thing that is found in the semen, are proteins and carbs. Proteins and carbs. All right, so these are embedded in the semen where you can find the sperm, and these perform different functions which I have mentioned already. All right, so let's look at what happens actually in capacitation before we look at acrosome reaction. Now, about 500 million sperms, about 500 million sperm is released into the reproductive tract of the female, right? But about 500, just 500, just 500 of the sperm are able to make it through to the fallopian tube or to the egg. Now, this is what we call 1 in a million. Okay, so one part in a million makes it to the sperm. But even at that, the 500, it is even at that out of the 500, it's only one that will fertilize the egg. All right, now let's talk about the sperm and capacitation now. Now, the sperm actually is made up of three different parts: the head, the neck, and the flagellum, which is the tail. Now, the neck part has mitochondria embedded in it. And what's the function of this mitochondria? It just makes the sperm strong. It gives the sperm energy. All right, you know, mitochondria is the part where energy is stored in ATP, in the form of ATP. All right, now let's look at what happens when capacitation occurs. Now, you need to understand that no matter how fast the sperm is, like I said in the first video, even if it gets to the oocyte, it cannot fertilize the egg until capacitation occurs. All right, so the fastest sperm can get to the oocyte within 5 minutes. But before this process, before fertilization will occur, capacitation must take place. And that's what we are going to be discussing now. All right. Now, the sperm has a helmet or a covering, and that covering is called the acrosome. Let's label that the acrosome. The acrosome. Now, on the acrosome, because it has to move through different parts before it gets to the fallopian tube, the acrosome is actually covered with proteins and carbs. Proteins and carbs covers the acrosome. All right. Now, not just that protein and carbs cover the acrosome. In basic biology, we know that the human cell, the cell membrane of the human cell, is made up of phospholipid layers. And we know what lipids are. They are just oils. They are unstable. They are not rigid. So something actually makes the cell membrane rigid. Now, that is what we call cholesterol. So we have cholesterol sitting on the cell membrane of the sperm. Sitting on the cell membrane of the sperm. And you may be wondering where this cholesterol is coming from. Remember when sperm are produced in the seminiferous tubules, what happens is that the sperm is produced, the type of cell that actually produces the cholesterol sits outside, and that's what we call the Leydig cells. Okay, now these cells produce this cholesterol. So that's where this cholesterol is coming from. Now, when this wants to undergo capacitation, something interesting happens. The first thing that happens is that the sperm has to get rid of the proteins and carbs. Now, capacitation is just an act of cleansing the sperm head, just cleansing the head. All right. So the first thing that happens is that the proteins and carbs will be off. It sheds off the proteins and carbs. It sheds off the proteins and carbs. That's the first process that occurs. Now, when that happens, we are left with just cholesterol. We're left with just cholesterol. Now, the next thing that happens after the proteins and carbs are shedded away, the cholesterol has to also leave the cell membrane so that the cell will be able to join with the oocyte. So this is what happens. The cholesterol starts leaving. That's the second thing. So when the cholesterol has left the cell membrane, what happens? The cell membrane becomes very vulnerable, very weak and loosed. All right. So it wants to give out everything it has. Okay. Now, this whole process of removing proteins and carbs, and removing cholesterol, making the cell very weak, making the cell membrane very weak, is what we call capacitation. So this process must occur before the sperm can fertilize the egg. And this, even though the father sperm can take about 5 minutes to get to the oocyte, this whole process of capacitation takes about 7 to 10 hours. 7 to 10 hours. All right. So now let's look at the next thing now, which is acrosome reaction. And the first thing that happens here is that after the cleansing of the sperm hairs, something happens. There will be an influx of calcium. There will be an influx of calcium. So calcium enters the cell. So this influx of calcium will cause a lot of things to happen. Okay. Now the first thing that will happen, or one of the things that will happen when calcium gets into the cell, is that it will trigger the mitochondria. Now, when it gets into the mitochondria, enough energy will be released. And when that energy is released, when that energy is released, it has an effect on the flagella, the flagellum, rather. And what this does is that it increases hyperactivity. So the flagella begins to whip as fast as possible. That helps it to run as fast or swim as fast as possible to the oocyte. Now, the next thing that happens after that is that these enzymes inside, remember the acrosome actually is an extension of the Golgi body. I know what the Golgi body does. It packages products, packages things in the cell, packages enzymes. So this is just an extension of the Golgi body. All right. So now in the acrosome, you have enzymes. These are the enzymes that will make sure that fertilization occurs. And this is what happens. There are two basic enzymes in the acrosome, and they are called hyaluronidase and acrosome. Now, hyaluronidase breaks down carbs. So this breaks down carbs. Meanwhile, this breaks down protein. So it's a protease that breaks down protein. Meanwhile, this breaks down carbohydrates. Now, what happens here is that remember that this cell, the first thing that will happen even before the enzymes are released is that there will be a receptor. There will be a receptor here. Now, what would this receptor do? It has a function to play, but we are going to talk about that very soon. Now, these enzymes are released. When these enzymes are released, what happens is that the enzyme begins to break down these granules that encoats or that coats the surface of the oocyte. So it begins to break down the granules. Now, these granules, when broken down, something happens. The sperm begins to join to with the oocyte. Now, this happens this way. So the sperm grows to join the oocyte. Remember, this enzyme will break down the granules, will break down this part, break down this part here also, and join here. Now, when that joining occurs, something interesting happens. Something interesting happens. Now, the joining will trigger a particular type of granules or cells inside the oocyte. Now, what this does, this is called the cortical granules. Now, what these granules do is that they serve as a security system of the oocyte. And so when the first sperm is able to destroy or break down the proteins or the carbs in the granules or break down the granules, it joins. The moment this happens, this remember these are they are the receptors are here already. Okay, receptors are here already. Now, what happens is that these cortical granules immediately shut down the zona pellucida. Now, this part is known as the zona pellucida. This part is known as the zona pellucida. So what this does is that the cortical granules release its substance into this first area, when it releases the substance into this first area, what that does is that it begins to shut down every part of the zona pellucida. And so what does this mean? This means that even if we have all that, remember we have around 500 sperm cells that are trying to fertilize the egg at once. Now, this means that the moment this shuts down, the whole of the 500 sperm cells goes off. And that is because the receptors that that will take the cells will be shut out. So all the receptors embedded in this part will will go off, so that this process of um, breaking down these granules to fuse will not occur. Okay, so the moment this senses that this the first sperm has been fused, these cortical granules produce the enzymes that shuts down the zona pellucida. And when that happens, no other sperm cells can fertilize the ovary. Okay, and so this is just what happens. Now when this happens, the next thing that happens is that the genetic material now the genetic material moves. So you have genetic material moves into the oocyte, merging with that of the oocyte. All right, so this is just what happens in acrosome reaction. So a quick run through: sperm is released, it runs. Before it gets to the oocyte, it must have undergone capacitation, which is the cleansing of the sperm head, removal of protein and carbs and the cholesterol, okay, which helps it to maintain its rigidity. Okay, so when the cholesterol is removed, the cell becomes very weak and is trying to empty things. We have a calcium influx that increases hyperactivity of the flagellum. Now, when that happens, the sperm tends to move faster. Now, when it gets to the the part the oocyte, it has to fertilize. So this occurs. We have the enzymes trying to break down the gates or the granules of the oocytes. Now, when this happens, it breaks it down. This part of the cell fuses with that of the oocyte and releases the genetic material after the cortical granules have shut down the zona pellucida. Okay, so this is just what happens in acrosome reaction. So thank you and see you in my next video.